students, and welcome once again to the e-learning portal of Palugat Mahila Mahavidyalaya. In my previous class regarding Marchantia in section 1, we were talking about the general vegetative structure of Marchantia. We were also looking about the mode of asexual reproduction in Marchantia. Today in my class we will be talking about the alternation of generation in Marchantia. But before this, I want you to recall whatever we have been talking about in the previous classes. Things like the haploid generation, things like the type of gametophyte the Marchantia has. I prepared few slides and I want you to go through them carefully and we will discuss about it later. Please follow the slides.
The fertilization in Marchantia is of interesting type. Fertilization takes place when a male and a female thalli grow near each other. As I've already mentioned, Marchantia is mostly a dioecious species. That is, there is a separate male and a separate female plant. Water is very essential for fertilization. As I have already told you in my introductory classes, bryophytes are known as amphibians of plant kingdom. That is, water is very essential for their reproduction. When a mature archegonium is developed, the neck of the archegonium is directed upwards on the dorsal surface of the disc of the archegoniophore. In the mature archegonium, the venter canal cells in the central region and the neck canal cells on the outer side of the archegonium disintegrate and forms a mucilaginous mass. This mass absorbs water, swells up and comes out of the archegonial mouth by pushing the cover cells apart. On the other hand, on the anthridiophore, the anthrizoids are splashed by raindrops. They may fall on the nearby female receptacle or they may swim in the film of water and reach the female receptacle. In all the cases, the fertilization is only possible if both the receptacles are surrounded by water. Many anthrizoids enter the archegonial neck by chemotactic response and reach up to egg. That is, there are chemical substances present in the mucilaginous mass that will attract the anthrizoids towards the egg. And this mechanism of fertilization is called splash cup mechanism, where the splash of raindrop will promote the transfer of anthrizoids from the anthridial filament to the archegonial filament. One of the anthrizoids penetrates the egg and fertilization is effected. The fusion of both male and female nuclei will result in the formation of a diploid zygote, also known as oospore. Fertilization ends the gametophytic phase, whereas formation of a diploid zygote is the starting of the sporophytic phase. The zygote will remain attached to the female gametophyte.
Now we have studied about the archegoniophore, the anthridiophore. Now let us look into the mechanism of fertilization in Marchantia. It is very important that both male and female gametophore will be close to each other while they are growing. Since Marchantia is a dioecious plant, that is, male and female sex organs are present in different thalli. So at the time of fertilization, water is very important. Remember my introductory classes where I have told you that water is important in case of bryophytes. That is why bryophytes are known as amphibians of plant kingdom. In this case, the male reproductive organ, the anthridiophore disc, will be like this. A drop of water will come and it will splash on the anthridiophore disc, and with this force, the anthrizoids present here will be carried with this drop of water to the female gametophyte or the archegoniophore. Now these anthrizoids, as I told you, they are motile in nature. They can swim in water. As I told you, these anthrizoids can swim in water and they are attracted by the chemicals that is produced by this archegonium. Now with the help of the rainwater, with the splashing of the rainwater, the anthrazoids are carried or transferred from the male anthidiopore to the female archegoniophore, and then ultimately the fertilization takes place. This type of fertilization that is aided by a splash of water is known as splash cup mechanism. So it is very interesting to see in bryophytes, if the water is not there, if the temperature or the climatic condition is dry, then bryophyta will never go for sexual reproduction. The most common mode of reproduction is always vegetative, but under favorable condition. As you can all see, water is so very needed for fertilization. Under favorable condition, Marchantia will show sexual type of reproduction. After fertilization, what are the post-fertilization changes that will ultimately lead to the formation of a diploid zygote and the sporophytic generation represented by 2n number of chromosomes will begin. Please follow the slides and we'll come back again later.
The post-fertilization changes and the development of sporophyte in Marchantia. After fertilization, the following changes occur simultaneously. The stalk of archegoniophore elongates. It becomes more and more long. Remarkable overgrowth takes place in the central part of the disc. As the result of this growth, the marginal region of the disc bearing archegonia is pushed downward and inward. The archegonia are now hanging towards the lower side of the neck, pointing downwards. The developing sporophyte is surrounded by three protective layers of gametophytic origin: calyptra, perigynium, and the pericytium. Zygote develops into the sporangium. The mature sporangium can be differentiated into three parts: the foot, the seta, and the capsule. The foot is a bulbous and multicellular structure. It is composed of parenchymatous cells. It acts as anchoring and absorbing organ. It absorbs the food from the adjoining gametophytic cell for the developing sporophyte. The sporophyte will never leave the gametophyte. The figure represented on your screen shows the development of a sporophyte after fertilization. The mature sporophyte shows a well-developed foot, a multicellular seta, and a bulbous capsule. The capsule contains spores and elaters. Elaters are specialized cells that will help the spore in dispersal. They are hygroscopic in nature and absorbs water. On the right hand side of your screen, you can see the microscopic image of the section of Marchantia sporophyte. The section also shows a multicellular foot that is attached with the gametophyte, the elongated seta, and a capsule containing numerous spores and elaters. Now, how is spore dispersed by the sporophyte? As the sporangium matures, seta elongates rapidly and pushes the capsule in the air through the protective layers. That is, now the capsule is very much visible outside the gametophore. The right capsule wall dehisces, that is, breaks from apex to middle by 4 to 6 irregular teeth or valves. Here the role of elaters is noticed. The elaters are hygroscopic in nature. In dry weather, they lose water and become twisted. When the atmosphere is wet, they become untwisted and cause the jerking action. Due to this, the spore mass loses and spores are carried out by air currents. Elaters are very important for the spore dispersal mechanism in Marchantia sporophyte. On the right hand side of your screen shows the structure of a mature sporophyte that has grown beyond the gametophore and is a well-developed structure. Here are the images of few of the sporophyte capsules found in different species of Marchantia. When the capsule is mature, it is clearly visible to the naked eye. These capsules When there is enough water in the environment, burst opens and the spores are liberated with the help of elaters. Under favorable conditions, the spores germinate immediately. In first year, the spore viability is approximately 100%. In Marchantia, 50% of the spore develops into a male thalli and 50% of the spore develops into a female thalli. I want you to note that the sporophyte contains spore mother cells which are 2n in number. After meiosis, the spore mother cells give rise to haploid spores. These haploid spores directly germinate to form a gametophyte. Dear students, here you can see the real time-lapse video of a mature sporophyte of Marchantia. You can see the mature capsule is liberating spores. The yellow cottony mass are the elaters. As elaters are hygroscopic in nature, they will absorb water and unwind themselves. This unwinding will cause the capsule to rupture and pushes the spore mass outside the capsule. Once the spores are liberated, they are carried away by the wind current or water to the nearby substrate. These spores are haploid in nature and directly germinates into a gametophyte.
Now let us look at the sporophyte of Marchantia. You must be following in the slides regarding the sporophyte. Now after fertilization, what happens? The female archegonium will contain the egg, and after fertilization the egg will be converted into a zygote. Zygote is a 2n structure, that is a diploid structure. This is the beginning of the sporophyte. Now what happens? The disc will expand and the sporophyte will continue to grow. The zygote, after repeated cell divisions, the details I have given in the notes that is been uploaded in the website. Now the zygote will ultimately form a structure known as the sporophyte. Now inside the sporophyte you will have certain cells which are known as spore mother cells, and these cells will ultimately undergo meiosis. The spore mother cell is 2n in number. After meiosis, the spore will become n in number. So there is a reductional division inside this structure which is known as the capsule. Now let us look in detail how the sporophyte of Marchantia will look like. This is the female thallus, female gametophyte. That the sporophyte is attached to the female gametophyte with the help of the structure known as foot, and the foot is embedded inside perigynum layer. There is a small long structure, cylindrical structure known as seta, and a round bulbous structure known as capsule, which is covered by a wall known as calyptra. Now inside the capsule you will see there are spore mother cells, or after cell division there is spores, and in between there are spiral elongated cells known as elaters. Now what are elaters? Elaters are actually spiral shaped cells, or they are long cells that are hygroscopic in nature, that is they can absorb water. Now when there is moisture in the surrounding, they will absorb water, and during the dry season they will release the water. So when they are absorbing and releasing the water, the elaters will twist and turn in a specific way. This will help the release of the spore. The capsule, when it is mature in the dry season, it will just burst open by the action of these elaters. That is why elater is very important for spore dispersal in Marchantia. This is the only structure in Marchantia that represents the sporophyte and is diploid in nature. So in case of Marchantia, the sporophyte is a diploid structure that contains foot, seta, capsule, as well as a structure that is always anchored, or that is always attached to the female gametophyte. This sporophyte will never leave the gametophyte. So that is why this sporophyte is not an independent sporophyte. So this is the type of sporophyte that we see. As in slides you must have seen different types of sporophyte occurring in the natural condition. Please follow the slides where we will be discussing about alternation of generation in Marchantia.
Now let us understand the alternation of generation in Marchantia. There are two different types of generation in Marchantia. One is the haploid or the gametophytic generation, and the other one is the diploid or sporophytic generation. The haploid generation or the gametophytic generation, represented by n number of chromosomes, is the dominant generation in Marchantia. In this generation, sex organs known as anthridiophore and archegoniophore are produced. Vegetative and asexual type of reproduction is also found in the gametophytic phase. Sex organs produce gametes to form a diploid zygote. With the formation of zygote, the sporophytic phase starts. The sporophytic phase, represented by 2n number of chromosomes, is initially represented by a diploid zygote that develops into a sporophyte. In Marchantia, the sporophyte will never leave the gametophyte and derives all the nutrient from the gametophyte through a specialized tissue known as foot cells. Marchantia sporophyte is represented by a foot, the seta, and a capsule. The capsule contains spore mother cells. The spore mother cells will undergo meiosis to produce haploid spores. These haploid spores on germination directly produces gametophyte. In this figure, we have clearly indicated the alternation of generation in Marchantia. As you can see on the left hand side of your screen, the germination of spore and development of a young gametophyte. The spores represent the beginning of the gametophytic phase. The male and the female gametophyte plants develop from the spores. The male gametophyte contains anthridiophore. The female gametophyte contains archegoniophore. Anthridiophore produces anthridia with sperm cells, and female archegoniophore produces archegonia with egg cells. After fertilization, a diploid zygote is formed. The zygote finally develops into a mature sporophyte. The sporophyte is attached with the gametophyte with the help of foot. The capsule contains sporogenous cells or the spore mother cells that will undergo meiosis. As a result, haploid spores are formed. These spores are liberated in the environment with the help of elaters, and finally the life cycle of Marchantia is completed. Here in the figure you can see the diagrammatic representation of the life cycle of Marchantia. Marchantia plants are dioecious in nature. Both the male and the female plants are separate. The gametophytic generation is the dominant generation, whereas sporophytic generation is the generation that is only represented by a mature sporophyte. The mature sporophyte contains foot, seta, and capsule. Gametophyte and the sporophyte are distinct in their morphology. That is why in Marchantia, two morphologically distinct phases, the haplophase and the diplophase, constitute the life cycle. This type of life cycle, which is represented by two morphologically different type of generations, where the gametophytic generation is a haploid generation and the sporophytic generation is a diploid generation, is known as heteromorphic haplo-diplobiontic type of life cycle. That is why Marchantia shows heteromorphic and haplo-diplobiontic type of life cycle.
Now Marchantia shows a very simple type of alternation of generation. Actually, when you see the Marchantia, the male gametophyte is the dominant structure that is represented by n number of chromosomes. There is a male gametophyte and a female gametophyte. Male and the female gametophyte will respectively produce archegonia 4 and anthradio 4. That will ultimately produce archegonia 4 will produce archegonium and egg. These are all haploid generation. Anthridium will produce sperm cells, and these are also haploid generation. After fertilization, the oospore is 2N, that is a diploid generation, that will finally produce the sporophyte that I already explained with foot, seta and a capsule. The spore mother cell, that is 2N, will undergo meiosis, the reduction cell division, and will produce n number of spores. After meiosis, the chromosome number is reduced to half, and then the 50% of spores will directly germinate into male and 50% of spore will directly germinate into female plant. This is the sexual reproduction. Only in sexual reproduction you will see the union of two gametes, the male and the female, and then that will ultimately produce a sporophyte. But when there is a vegetative reproduction, the asexual reproduction, the entire asexual reproduction is haploid in nature. You know about asexual reproduction by three modes of asexual reproduction: by gamete, by adventitious branches, as well as fragmentation. When you look at the entire life cycle of Marchantia, you will find two important things. That is, the gametophyte is represented by a simple thallus that will bear archegonia, or that will bear the anthridium. So this is the gametophyte. Now sporophyte is represented by a capsular structure that is attached to the gametophyte with the help of the foot. Now these two structures are morphologically different structures. That is why they are known as heteromorphic. Heteromorphic means different morphology. Now since they are also showing two types of generation, one is the haploid generation and another one is a diploid generation. That is why it is known as haplodiplobiontic type of life cycle. So in this case, Marchantia shows heteromorphic haplodiplobiontic type of life cycle. In my next class, we will be studying about Anthroceros. Anthroceros shows a semi-parasitic type of sporophyte, but here the sporophyte is not independent. The sporophyte cannot photosynthesize and it is fully parasitic or dependent to the gametophyte in case of Marchantia. Students, I hope you all are enjoying the classes, and I hope you all are also following the slides that has been made. Please don't forget to subscribe the YouTube channel, and also please call me, or we can share the views of further explanation in WhatsApp, and also download the study material from our website balubhattmmb.com, and hope to see you in the next class.